Hello friends and welcome to CEC Gurukul series. I am Dr. Jai Malik, Assistant Professor of Pharmacognosy at University Institute of Pharmaceutical Sciences, Panjab University, Chandigarh. So today I will be delivering a series of lecture in which we will discuss about various topics that are mentioned under Pharmacognosy and Phytochemistry syllabus for B.Pharm 4th semester as prescribed by Pharmacy Council of India. So in the series, in the first lecture, I will be discussing about what is pharmacognosy, what was its brief historical development, what is the scope of pharmacognosy, and in the subsequent lectures we will discuss what are crude drugs, herbal products, natural medicine, what are the sources, various sources of crude drugs, how these crude drugs are classified, what are the various systems of classification, what are their advantages and disadvantages, followed by the quality control methods of herbal drugs, their adulteration, different types of adulteration, different methods of adulteration with suitable examples. So to begin with the historical background of the pharmacognosy, we can certainly say that the plants exist on this planet much before the evolution of man, and perhaps the use of plants by man is as old as the man's evolution. This fact is further substantiated by the remains of medicinal plants that were found in the 60,000 BC old burials in the Chandigarh caves in Iraq. And in this picture you can clearly see that this is a reproduced model of a Chandigarh burial, and these Chandigarh caves gave the evidences for the first burials of the mankind. So once the man was evolved, and since plants were much before the man evolution, he started using plants to satisfy his primary need, that is hunger, at that time. And after satisfying his primary need, that is hunger, he started discovering other uses of plants. And the other uses of plants he discovered on the basis of experimentation, hit and trial, by observing animals, and by accidental discoveries. So during experimentation, the plants he found suitable to eat, he categorized them as edible, and those which were not suitable for eating, they were categorized as non-edible. Similarly, in hit and trials, various plants were used for different disorders, and those plants that were safe after eating, he considered them as safe, and those which were not safe, he considered them as toxic. Similarly, he also observed animals which were living in close vicinity of the early man, and he by observing animals, he saw that there are certain plants or there are certain plant products that can be consumed by the man, and there are certain plant products which cannot be consumed by man. And same was the case in the accidental discovery aspects. This primary knowledge or the empirical knowledge which he gained by experimentation, hit and trial, by observing animal, and he passed on this empirical knowledge from generation to generation, and with each generation some new knowledge was added, and this lead to the formation of a pool of knowledge. This collective knowledge laid the foundation of different traditional systems of medicine. This knowledge that was totally empirical, on experimental basis, experienced basis, and this collective knowledge laid the foundation of different systems, that is Ayurveda, which is our traditional system of medicine. Similarly, other traditional system of medicine such as traditional Chinese medicine system, Egyptian system of medicine, Japanese system of medicine, which is also known as Kempo, and Arabian traditional system of medicine, that is Unani. Besides this, many civilizations such as Greek, Romans, Egyptians were perished, but the traditional systems of medicine developed by them are still used in different parts of the world. And these systems, one thing was very common in these systems, that all these systems were based primarily on the plant-based products. No doubt certain animal drugs were also mentioned in these systems, but the number of animal drugs were very less. Now I will discuss briefly about the Indian tradition of medicine, that is Ayurveda. And Ayurveda is considered as one of the oldest system of medicine. Perhaps it is the oldest system of medicine, and it is generated by combination of two words, Ayur and Veda. Ayur means life, and Veda means science of life. So Ayurveda is basically science of life. And the empirical knowledge that was passed on from generation to generation was also used by various rishis in our culture, ethnic or mythological history, and these rishis used that empirical knowledge in writing the Vedas. And it is believed that Ayurveda has developed from Vedas, primarily Rig Veda and Athar Veda. And these are expected to be written around 3500 to 1500 BC. And these Vedas have descriptions of various properties, medicinal properties of number of plants. Further, in Athar Veda, there are hymns which mention about medicines like Acoramus calamus, which is commonly known as Bach, and Phyllanthus embellica, which is commonly known as Amla. So here we can clearly see that the medicinal plants were mentioned in our Vedas also. Now Ayurveda has three major writings. One is Charak Samhita, second is Shushrut Samhita, and third is Ashtam Hardayam Samhita. Charak Samhita was compiled by Agnivesha and is considered as treatise on medicine. Similarly, Shushrut Samhita was compiled by Shushrut, who is also known as father of surgery, and is considered as treatise on surgery. Ashtang Hardayam Samhita was compiled by Vagbhat and is compilation of both the knowledge of Charak Samhita and Shushrut Samhita. Thus, Ashtang Hardayam Samhita brings both medicinal and surgical diseases under one common umbrella. Now in Ayurveda, the medicine is prescribed after understanding the nature of the patient's body, and nature in Ayurveda they call it as Prakriti. Whereas in modern system of medicine, a drug is prescribed after understanding the disease. So this is the basic difference between the Ayurveda and in the allopathic or the modern system of medicine, where in Ayurveda they understand the disease as well as the nature of the patient's body, that is Prakriti, whereas in modern medicine system the main focus is on the disease of the patient. So if we want to give an example on that, if 2 or 3 people are having a particular disorder, so in a modern system of medicine, a doctor will understand that particular disease and will prescribe a medicine. So all the 3 or 4 patients can have a similar kind of medicine for that particular disorder. Whereas in Ayurvedic system of medicine, the Ayurvedic practitioner will first try to understand the nature of the body or the Prakriti of the body of the patient, and after that he will prescribe a medicine to that patient. So 2 or 3 patients can have a similar medicine, or they can have altogether different medicine for the same disorder. So Ayurveda, that's why it is considered as holistic system of medicine, which considers each and every body as a separate whole system, and depending upon the properties of those system, the medicine is prescribed in our traditional system of medicine. How the nature of the body of the patient is assessed. The nature of the body is assessed mainly on the basis of 3 dosha of our body, and these 3 doshas are responsible for controlling various aspects of our body, and these three doshas have been categorized into Vat, Pitta and Kapha. Vat initiates and promotes the biological activity responsible for the movements of the body, whereas Pitta is basically responsible for generation of heat and all the action relating to with the digestion, absorption, metabolism, pigmentation of skin, vision, intelligence and other mental functions. So we can say that if a person is having high metabolic rate, we normally say that it is having high Pitta. If a person is having low metabolic or digestion rate, we say that person is having low Pitta component. Third one is the Kapha, which is responsible for moistening of the ingested food, nutrition to the heart, mind and body tissues, perception of taste and lubrication of joints. So all these three dosha, when they are in harmony inside our body, then we say that our body is perfectly fine. And whenever these 3 doshas are out of synchronization or out of harmony, there is an imbalance between these three doshas, then our body will be suffering from one or some other disorders. So depending upon the 3 doshas, whether Pitta is high, whether Kapha is high or Kapha is low, depending upon the parameter, these parameters, the Ayurvedic practitioners prescribe the medicine. So similarly, as Ayurveda is primarily based on plant-based system, and a huge amount of knowledge has been contributed by Ayurvedic system on medicinal plants. Similarly, Chinese system of medicine has also contributed significant information on medicine plants. Though Chinese system of medicine, we all know that it rely on the yin and yang energy basis, but I will not go into detail of yin and yang. We will focus on its contribution to the medicinal plant area. So in Chinese system, Pen Ti Sao was a materia medica that was written by a Chinese emperor Shen Nuang in 2700 BC, and he compiled around 365 medicinal plants and is considered as the oldest records in China. Further, it was also believed that Emperor Shen Nuang used to test so many herbal drugs on himself before he compiled this information. So after Shen Nuang, it was followed by the inscriptions from the Shang dynasty that were written in 1766 to 1122 BC. In these inscriptions also, besides those 365 medicinal plants that were described by Shen Nuang, number of other plants were included and a document was compiled. Next in the list was Shan Hang Lung, which is known as Treatise on Acute Disease Due to Cold, and it was written by Chang Chun Chin in 142 to 222 AD. Now this is the era after the death of the Christ, and it is considered as the most important clinical manual of traditional Chinese medicine because it contains various herbal drugs and their preparation, their recipes, which can be used in acute diseases related to cold or infectious diseases. Now during 456 to 536 AD, Tao Hongjing's compilation, that is Commentaries on Herbal Classics, was released, and it has a description of around 730 herbs, their recipes, their characters, and where these drugs can be used. In this slide, you can clearly see the picture of Shan Nuang and Chan Chung Ching, who were considered as the persons who laid the foundation of the traditional system of medicine. The third image is of Abaris Papyrus, which is an Egyptian document and still existing document. Besides Indian and Chinese traditional systems of medicine, Egyptians, Greeks and Romans also played a significant role in imparting knowledge on medicinal plants. Huge amount of knowledge we all know that Egyptian and Greeks also impart that knowledge, and this knowledge is still being used in various parts of the countries. Aber Papyrus, the image which you saw in the last slide, that was written in around 1550 BC, and it is an Egyptian collection of around 800 prescriptions containing 700 drugs. So you can see that Abaris Papyrus was the most elaborated document on the medicinal plants and its prescription, and it is considered as one of the most complete and existing medical document of the Egypt, and this document still exists in Egypt. It has numerous medical texts, hundreds of recipes classified by disease, and anatomical monographs. So till date we were just having various writings of the traditional system of medicine which describe only the plants and their uses. Aber Papyrus was the first document in which the plants were written and classified on the basis of diseases. Hippocrates, whom we know as father of medicine, he was taught under the Egyptian priest doctors, and he wrote a treatise of about 300 to 400 medicinal plants, including common garden mint, which is also known as Mentha spicata, opium, which is known as Papaver somniferum, rosemary, Rosmarinus officinalis, sage, Salvia officinalis, and verbena, that is Verbena officinalis. Hippocrates was followed by Theophrastus, who was a Greek scholar and a student of Aristotle, and Theophrastus was known for his writing on the plant kingdom, and we all know that Aristotle was the person who was known for his writing on the animal kingdom. So Theophrastus also contributed large amount of information about the classification of the plants into various genera and families. Third in the list is the Dioscorides. He is also known as father of pharmacognosy, and he described various medicinal plants and their properties. He published 5 volumes of book on pharmacopoeia entitled De Materia Medica in 78 AD. Now, to honor his work, one of the genera known as Dioscorea has been named in the honor of Dioscoroids. And Dioscorea is the genus from which we obtain various plants of commercial importance. Now, this encyclopedia work described more than 600 medicine plants, including their name, synonym with pictures, habitat, botanical description, drug properties, medicinal use, collection and storage instructions, adulteration and method of detection, and veterinary uses. So the document written by Dioscoroids was so elaborated that it almost covered all the parameters that are studied in the modern pharmacognosy. And you can see that this Materia Medica was written way back in 78 AD. So hence we can imagine the type of elaborated work that was given by Dioscoroids, and that's why he is known as father of pharmacognosy. He gave the pictures, the habitats, botanical description, how the plant morphological characters are there, drug properties, what are the pharmacological effect of the drug, where these drugs can be used, how these drugs, besides their medicinal uses and properties, he also mentioned how these drugs have to be collected and storage, because we all know that the collection and storage aspects are important in case of herbal drugs. So Dioscoroids also elaborated on the collection and storage instructions of those herbal drugs. Further, he also made an important point about adulteration of these herbal drugs. We all know that adulteration is an activity that hampers the quality of the herbal drug, and that is why he basically pinpointed the adulteration aspect of herbal drugs. What are the various adulterants that can be used for different drugs, and how these can be, not only he told about the adulteration, but he also emphasized on the methods which can be used for the detection of those herbal drugs. And besides the medicinal uses of herbal drugs, he also focused on the veterinary uses of these drugs. After Dioscoroids, Galen was the person who basically slightly modernized the herbal medicine, and he discovered various methods of preparing different preparations of the plants and animal drugs. So Galen was the person who gave different preparations and their method of preparation of those formulations earlier that were used. Say for example, herbal drugs were mainly used as extracts or as powders. So Galen was the person who gave the method for preparation of these formulations, and to honor Galen, the traditional formulation which were known as Galenicals were named after him. Thus we can say that different civilization contributed to the knowledge of the plants and their medicinal uses, and with the passage of time and gradual development of scientific knowledge, scientists start studying plants mentioned in those traditional systems systematically and scientifically. So after that knowledge was gathered in those traditional systems of medicine, and as the knowledge of man, its scientific ability improved, he start using that knowledge of traditional system of medicines by studying those plants mentioned in those systems more systematically and scientifically to validate their traditional uses. The knowledge thus obtained and the knowledge collected from different sources, these sources can be traditional systems of medicine, these sources can be the experience-based knowledge of different people or ethnic groups. These knowledge was compiled in the form of books, and these books were earlier known as herbals. Later on, these herbals were renamed as Materia Medica. So Materia Medicas were an important contribution in the field of pharmacognosy, because these are the books which were related to the knowledge on medicinal plants, their properties, their uses. Apart from their uses, they had a focus on their morphological identification of the plants as well. So in this slide you can clearly see a image of an herbal, and which were renamed as Materia Medica, and in the picture you can see the image of a plant given and its description on the side in the herbals, and in Materia Medica its pharmacological action or its action on human body was also included. These are some of the oldest medicinal plants books. One is Ortius Santaris, that was written in 1491. Second one is De Historia Stirptum, that was written in 1542 by Leonard Fuchs. And the third one is a New Herbal, that was written in 1551 by William Turner. These are the three books which are still available and are preserved in various libraries. Now, with the gradual development of understanding on the plants and their medicinal value, a new subject was born, and this subject we named as Pharmacognosy. So, we can say that Pharmacognosy was a subject that was based on the knowledge given in the traditional systems of medicine, or the knowledge that was along with the various ethnic groups or the tribal people. So, all that knowledge gave rise to a subject which we call it as Pharmacognosy. Now, the term Pharmacognosy was coined by an Austrian physician, Johann Adam Schmidt, and he mentioned this the term in his manuscript Lehrbuch der Materia Medica, which was published after his death in 1811. So, Schmidt was the first person who mentioned the term Pharmacognosy in his manuscript, though he died before his manuscript was published, but he is considered as the father who gave the term Pharmacognosy. Later on in 1815, C.A. Sidler also used this term Pharmacognosy in his book Analytica Pharmacognostica. Now, this was in brief about how the subject was developed, how the subject was born. Now, what is basically Pharmacognosy? Etymologically, if we see that Pharmacognosy is a term that is derived from two Greek words, pharmakon and gnosis, which is knowledge, or gignoso, to acquire knowledge. So, in short, Pharmacognosy is a combination of two words, pharmako and gignosko, that is to acquire knowledge or to study about a drug. And Pharmacognosy was considered as a specialization dealing with the study of crude drug, mainly of plant origin. Later on, various modifications were done in the definition, which we are going to discuss in the subsequent slides. So, earlier it was just considered as a study of crude drugs, primarily of plant origin. And as already discussed, that earlier the plant drugs were mainly used in the form of powders, extracts, galenicals, and so on. But the changes started to begin in 19th century, when the isolation from these plant preparations were done. Now, these extracts were used to isolate various chemical constituents that were present in the plant, and various compounds were isolated during the 19th century, such as morphine, that was isolated in 1803, strychnine was isolated from Strychnus nux vomica seeds in 1817, quinine and caffeine were isolated in 1820 from cinchona species and tea respectively, nicotine was isolated in 1828 from tobacco plant, and atropine was isolated from belladonna in 1833. Now, these isolations encourage the scientists to work in the direction of isolating more and more compounds, so that new compounds can be identified and their biological properties can be studied. So, another major change that occurred during 19th century was the introduction of microscopy for identification of plant drugs, and for many years Pharmacognosy remained confined with the microscope based methods, and this introduction of microscopy was a path breaking introduction in this field, because earlier the plants were basically identified morphologically by just seeing their macroscopic characters, but many a times it happened that 2 or 3 plants were morphologically quite similar, their species are quite similar, they cannot be identified, and that lead to the adulteration, or you can say adulteration without any intention, because those plants were not clearly identified, and sometimes that lead to certain toxic effects in the human body. And now the introduction of microscopy lead to the more authentic identification of the plant drug as compared to those that were done on the basis of macroscopic study. So, this was a major introduction in the field of pharmacognosy during 19th century. Now, during 1850 to 1890, the subject went into a dormant phase because though the scientists were working in the direction of isolation of newer and newer components, phytoconstituents, but nothing new was coming out of, as these phytoconstituents were very difficult to elucidate their structures and to identify the exact structure of those compounds. So slightly due to this disadvantage, the subject went into the dormant phase. But in 1899, when aspirin was launched, we all know that aspirin is acetyl salicylic acid, and the salicylic acid that was used for preparing aspirin was obtained as a lead molecule from the plant Salix alba. So the marketing of aspirin 1899 by Bayer was the major boost again for the subject pharmacognosy, as now scientists start looking in a different aspect on this subject, that the plant products can be marketed and they can act as novel drugs for various disorders. After that, we all know that any subject, if it is there, it does not remain static, and so was pharmacognosy. So pharmacognosy also was not static after the launch of aspirin. And launch of aspirin laid down the pathway for the change in the traditional pharmacognosy. Earlier, we all know that the pharmacognosy was dealing with the plant-based drugs, their macroscopy, and how where these drugs can be used. These drugs can be used in different diseases. We were just studying that. But by the launch of aspirin, the traditional pharmacognosy was going to change, and it started changing during the 20th century. Now, the transition of the traditional pharmacognosy to the modern pharmacognosy started way back in 1930s. And how this transition happened? This transition happened with the three major discoveries. First was that pharmacognosy then became a multidisciplinary approach or subject. Various other disciplines like chemistry, biochemistry, pharmacology, and analytical chemistry get hand in hand with pharmacognosy. People from chemistry start studying plants for their various chemical constituents. What are those constituents, their identification, their structure elucidation. Then biochemistry and pharmacology people start working on plants, and they start discovering how these plants act on the body, by which they show their medicinal effect in the body. Analytical chemistry people start studying about the various developing various analytical methods by which they can determine the quantity of those active constituents in the plants. Further, the second major discovery was the chromatographic techniques that were developed during the 20th century and after 1930s. So these chromatographic techniques revolutionized this subject pharmacognosy, and at present chromatography is an inevitable part of plant drug studies. Chromatography led to further authentication or identification of plants. Various chromatographic profiles were developed of different medicinal plants, which further helped in the identification of the medicinal plants. In addition to that, in vitro bioassays were developed by the end of 20th century. And when these bioassays were developed, they opened the door for fast screening of various herbal drugs for various disorders. Now people don't have to go for animal studies or clinical studies directly on man or animals. They had in vitro bioassays, and they can screen number of medicinal plants in a very short time. Thus all these changes lead to the change in the definition of pharmacognosy. Then in 1985, Wallace defined pharmacognosy as a distinct department of pharmaceutical science dealing with a systematic study of structural, physical, chemical, and sensory characters of crude drugs of animal, vegetable, and mineral origin, including their history, cultivation, and collection. So until now we were studying medicinal plants just on the basis of their morphology and their effect on the body, but with the advancements in microscopy and chromatography and other allied branches coming along with pharmacognosy, the definition was changed, and now we are also studying the chemical and sensory characters of the crude drugs, that not only from plants but also from animal and vegetable, animal and mineral origin also. In addition to these, we also start studying about the cultivation and collection of the plants. Earlier we were mainly focused on the wild sources of the drugs, but since their importance was increased during the 19th century, the drugs were also being started cultivated. And what are the various methods of cultivation of those herbal drugs or medicinal plants, and how they should be collected? More emphasis was also given in the 20th century. Later, American Society of Pharmacognosy described pharmacognosy as the study of physical, chemical, biochemical and biological properties of drugs, drug substances or potential drugs, or drug substances of natural origin, as well as search of new drugs from natural sources. So apart from physical and chemical and biological properties, new things such as biochemical studies and the role of plants in new drug discovery was also highlighted in this definition by American Society of Pharmacognosy. So these definitions keep on changing on over a period of time in 20th century. Later, A. Douglas Kinghorn, who is current editor-in-chief of Journal of Natural Products, defined pharmacognosy as pharmaceutical science that deals with the discovery, characterization, production and standardization of drugs of natural origin. So we can clearly see that discovery means what is their use, what is their medicinal property. Characterization involves the structural elucidation of the chemical components that are present in the plant. Production, how they can be cultivated, and collection, all these aspects were there in the previous definition, but the new word in the definition was standardization of the drugs of natural origin. So in the 20th century we came over another problem, that is adulteration of those herbal drugs, because till the beginning of the 21st century, most of the herbal drugs were obtained from the wild sources, and lot of adulteration was there in those herbal drugs. So to maintain the quality and safety of those herbal drugs, standardization became the buzzword and the most important aspect of herbal drugs or plant drugs. In early part of the 19th century, so we can say that in the early part of the 19th century, the term pharmacognosy that was coined, and by the last half of the 20th century, pharmacognosy was evolved from being a descriptive botanical subject to one having a more chemical focus, embracing a broad spectrum of disciplines including botany, zoology, ethnobotany, marine biology, microbiology, herbal medicine, chemistry, biotechnology, phytochemistry, pharmacology, pharmaceutics, clinical pharmacy, pharmacy practice and so on, and thus pharmacognosy became a highly interdisciplinary science. So earlier the pharmacognosy was more or less a botanical subject in which we just go for the macroscopic characters of the plant, how the plant have botanical characters, how they can be classified in various kingdoms and genera. So this was a major aspect of pharmacognosy. Later we also start studying their biological effect, but by the end of 20th century it became a multidisciplinary subject, and people from other areas like biotechnology, pharmacology, pharmaceutics, they all start working on plants, and they start working on the new formulation. Pharmaceutical people start working how new formulations can be prepared. Clinical pharmacy people start working how these plants can be used clinically, and chemistry people start working what are the various constituents and how their structures can be elucidated. So overall it becomes a very interdisciplinary or multidisciplinary subject. Now, what changes occurred during the 21st century, that is the current century? So, enormous developments took place in the 21st century, and emphasis was mainly on the analysis, standardization, biological testing and collaboration of pharmacognostical research. Now, analysis became an important aspect of pharmacognosy to maintain the quality of the herbal drugs. So, analysis and standardization go hand in hand to maintain the quality as well as safety aspects of herbal drugs. Further, the biological testing aspect also improved, and various modern methods of testing the effects of plants were developed. Various in vitro methods, various in vivo methods were developed. Various bioactive markers were identified from the plants, and various biomarkers of various diseases were developed, which further helped in the biological testing of the medicinal plants. And the fourth one is the collaboration of pharmacognostical research. Now, pharmacognosy people were not just working on the medicinal plants, their botanical aspect, but they start collaborating with the people from different branches such as pharmacology to understand their mechanism of action, how they act inside the body. Then they also start collaborating with the biotechnological people, how to improve the quality of that medicinal plant. They start collaborating with the agricultural people, how to improve their cultivation methods so that the quality of the medicinal plants further improve. They also start collaborating with the chemistry people to elucidate the structures of the chemical constituents that are present in the plant. So all these developments start taking shape in the 20th century. Besides these, high throughput screening and hyphenated techniques were also developed during the 21st century that leads to the rapid evaluations of herbal drugs, and also structural elucidation of the chemical constituents that were present in the plant drugs. Then DNA barcoding or sequencing for the identification, authentication and checking the adulteration. Now, DNA sequencing was an important parameter that was included in that has been included in pharmacognosy, and this barcoding or DNA sequencing has helped a lot in authentication of the herbal drug that is being used by various industry for preparation of various herbal formulations. Because many a times it happened that there are two species that are morphologically or microscopically quite similar, and it's very difficult to differentiate between the two species. No doubt chromatographic techniques and their profiling has helped in differentiating the species as well, but many times it's very difficult because the concentration of the components, we all know that, vary from species to species, and they also varies on the basis of the geographical location of the plant. So DNA sequencing has helped a lot in giving the authentic identification of the plant drugs. And the last is the plant genomic and development of plant genomics and metabolomics. In addition to genomics and metabolomics, proteomics is also an upcoming field in the plant pharmacognosy. In genomics, we study about the various genes that are responsible for the production of various chemical constituents or various biologically active chemical constituents in the plant. And in metabolomics, we basically study the combination of the metabolites, which are known as metabolomes, and these we also study what are the various pathways and how these metabolomes or metabolites are produced inside the plant. And in proteomics, we basically study what are the various proteins and what are the various enzymes that are responsible for the production of these biologically active or other metabolites in the plant. So we can say that today pharmacognosy has become an important branch of pharmacy that includes the scientific study of structural, physical, chemical, biochemical and biological property of crude drugs, and search for new drugs from plant, animal and mineral sources. So thus in pharmacognosy today we basically study the plant drugs on the basis of their morphological characters, microscopical characters, their chemical compositions, how they act on the body, what are the biochemical changes these plants lead inside a human body, and how these can be prepared, how different formulation of these drugs can be prepared, and how these drugs can help us in discovering newer drugs. And these crude drugs we obtain from plants, animal and mineral sources. So today herbs can also be turned into commercial products, and pharmacognosy is playing an active role in the discovery, characterization, production and standardization of natural drugs. So in the 21st century, rather in the last by the end of the 20th century also, various herbal products were there in the market, and now we can say that these medicinal plants or herbs can be converted into commercial products. And pharmacognosy plays an important role because a pharmacognosist has to characterize that plant drug, has to standardize that plant drug, has to develop various improved methods of its production. So pharmacognosy is now not only the botanical study but covering all the aspects of pharmaceutical sciences. Now, besides the study of plants as drugs, now pharmacognosy also involves the study of plants that are used as pharmaceutical aids. We all know that various pharmaceutical preparations that are being manufactured by pharmaceutical industry, they also involve the utilization of various pharmaceutical aids such as coloring agent, emulsifying and suspending agents, and various plants are also used as coloring or emulsifying or suspending agents. Coloring agents have an examples curcuma, which we commonly known as haldi, saffron, which we commonly known as kesar, similarly beetroot, chakundar. These are also used as coloring agent in the manufacturing of various pharmaceutical products. Similarly, various gums such as gum acacia, tragacanth and isabgula are used as emulsifying and suspending agents in pharmaceutical industry. Various plant drugs which are having essential oils are used as flavoring agents, like mentha, lemongrass, eucalyptus. All these are used as flavoring agents in various preparations. These plant products are also used as diluents and bulking agents or fillers. For example, cellulose, lactose, sucrose, and these are being used in the preparations of pharmaceutical formulations like tablets and capsules. Plants products can also be used as disintegrants, which are widely used in tablets. For example, CMC, that is carboxymethyl cellulose. Plants are also used as natural sweeteners such as licorice and stevia. They are also used as adhesives. Guar gum, amylase and caraya gum, beeswax, cocoa butter and theobroma oil are used as solidifiers. So we, apart from the plants that are used that are medicinally active, we also study the plants that can be used as pharmaceutical aids in pharmacognosy. We also study about various fibers that are used to prepare surgical dressings and filter medias. For example, cotton, jute, they are used for preparation of various surgical dressings. We also use diatomite, which is used for preparation of filter medias. Besides medicinal plants, pharmaceutical aids, pharmacognosy also involves the study of various poisonous, hallucinogenic and teratogenic agents, and the common examples of such plants is Abrus precatorius, Atropa belladonna, Cannabis sativus. Cannabis sativus has been used since ages as a hallucinogenic plant. Datura stramonium, Ipomia purpurea and Salvia species. Thus, today pharmacognosy can be defined as a systematic, multidisciplinary study of crude drugs obtained from plants, animal and mineral sources. Now, the term study includes the study of their history, biological source, macro and microscopic characters, cultivation and collection practices, ethnobotany, ethnopharmacology, phytochemistry, biological uses, molecular analysis and quality control method to assure the quality, potency, purity and safety of crude drugs, and how these can assist in the new drug discovery. So we can clearly see the difference in the definition that was given during the 19th century when pharmacognosy was born. At that time it was just a study of crude drugs, mainly of plant origin, and today in 21st century pharmacognosy has become a multidisciplinary study which involves the study of their biological source, from where these plant drugs are obtained, their macro and microscopic characters, their cultivation and collection practices, how these can be cultivated and what are the correct collection practices that should be followed to maintain the quality of these drugs. Then the ethnobotany and ethnopharmacological aspects of these drugs. Ethnobotany and ethnopharmacological aspects involve the information that belongs to certain ethnic or the tribal people, because these tribal people have number of have lot of information on the medicinal plants that is being used by their generation to generation, and those plants have not been scientifically validated till date. Number of plants that are being used by those people have not been scientifically validated. So we also consider today in pharmacognosy that ethnopharmacological aspect of those plants. We are also considering phytochemistry as part of pharmacognosy, because in this also, in pharmacognosy today, we also study about the type of chemical constituent present in the plant, their structure elucidation, their classification, and how these chemical constituents are important for those plants. Then how these plants are used medicinally, what are their medicinal properties. We also study that. Molecular analysis and quality control methods to assure quality, potency, purity and safety of the crude drugs. Now quality, purity and safety have become an important aspect in the usage of medicinal plants, and pharmacognosy also today also deals with all the three aspects, that is quality, purity and safety of crude drugs. And besides all these factors, in pharmacognosy today we also study how the plant drugs can assist in the discovery of newer and safer drugs. So in if we summarize what we have studied, discussed till by this time, is that use of plants has been an integral part of human development, and the day we were evolved on this planet, we are using plant. Initially we were using plants for fulfilling our primary need, that was hunger, and later on with experimental or hit and trial methods, we discovered other uses of plants. Then this empirical or experimental knowledge was passed on from generation to generation, that help in the development of various traditional systems of medicines. So all that empirical or experimental based knowledge was gathered from one generation and was passed on to the next generation, and with each passing generation certain new knowledge was added, and this empirical or experimental knowledge laid the foundation for the development of various traditional systems of medicines. And the major one thing was quite common in all the traditional systems, that they were primarily based on plant based medicines. That traditional knowledge that was compiled in the form of herbals, which were renamed as materia medicas, and later on with the development of scientific knowledge and temperament of scientists, a new subject, that is pharmacognosy, was born, and the term pharmacognosy was coined by Johann A. Schmidt in his book that was published after his death in 1811. And pharmacognosy is basically a combination of two Greek words, that is pharmakon, which is known as drug, and gignosko, to acquire knowledge. So pharmacognosy is to acquire knowledge of a drug. So various changes occur during the 19th century, and the changes begin with the isolation of pure phytoconstituents from the plant, and incorporation of microscopic studies in pharmacognosy to study the characters of the medicinal plants, and to help in the identification of those plants. And by the end of the 19th century, pharmacognosy got a major boost when aspirin was launched by Bayer in 1899. So with the launch of aspirin, the outlook of the scientists toward pharmacognosy changed altogether, and they start working in the direction of discovering various commercial products. So later in 20th century, it became a multidisciplinary subject from a single outlook subject, that is the botanical and medicinal aspect of the plants. We shift to the multidisciplinary subject in 20th century, and stress was being laid on the correct identification, biological evaluation and analysis of herbal drug using various chromatographic techniques. So now in 20th century, more and more evaluation aspect was there of herbal drugs, whether that evaluation was biological or analytical evaluations were there. We were more concerned about how these plants act on the body, as well as how these the quality of these plants can be maintained. What are the various parameters that can be evaluated to give a particular parameter for the quality. Then in 21st century, enormous development occur in pharmacognosy, and today lot of emphasis is being given on the standardization of herbal drugs, because we know that herbal drugs are mainly collected from the wild sources, and very less percentage is obtained from the cultivated sources. So whatever we are collected from the wild sources, we sometimes we don't know whether we are collecting the actual plant or not, and that sometimes lead to certain toxic effects in the body. So lot of emphasis is given on the standardization of herbal drugs. So one aspect of standardization is the identification of the herbal drugs, and second is to maintain the quality, and that quality is maintained by the analytical aspects of the herbal drugs, and analytical chemistry people are also coming hand in hand with the pharmacognosy people for that. Then toxicity and safety profiles of herbal drugs are also being monitored, and they are prepared by various agencies across the globe, so that these herbal drugs can be utilized to their fullest. DNA barcoding and DNA sequencing also helped in identification of the plants, and this gave a foolproof method for identification and authentication of the plant raw material. Then in 21st century, plant genomics and metabolomics and proteomics entered into the field, and they revolutionized the pharmacognosy subject. Now plant genomics, as we already discussed, that it deals with the study of genes, the DNA structures, how these genes, what are the various genes that are responsible for the properties of the plant, what are the various genes that are responsible for the production of the secondary metabolites or the biologically active chemical constituents in the plant. In proteomics, we study about the various protein compositions, what are the various enzymes that are responsible for the biosynthetic pathways or biosynthesis of secondary metabolites. And in metabolomics, we study about the various metabolites that are produced inside the plant. So all these changes basically happened or were incorporated or were the additions in the subject pharmacognosy. So pharmacognosy is an important part of new drug discovery process, and today traditional drugs are getting shape of marketed products. We all know that allopathic medicine or the synthetic drugs are being used today, and many people feel that they possess various side effects, and that's why man is once again moving back to nature, and he is looking for more safer drugs for usage of various disorders, and that's why plants are becoming an integral part of drug discovery process. And traditional drugs, which we commonly use as extracts or powders, are getting shape of marketed products. We know that various herbal tablets, herbal capsules, herbal syrups are there in the market, and they are being used by man, and as per WHO, around 80% of the world population still rely on the herbal products. So thus we can say that this was an overview how the pharmacognosy changed its shape from its birth in 19th century to the 21st century, where the herbal drugs are being used as commercial products, and they are being produced in the market. So in the next lecture we will discuss about the scope of pharmacognosy.